The overall goal of this method is the delivery of RNA interference triggers in Anopheles gambiae to initiate gene knockdowns that begin during the pupal stage of development.
This method can help answer key questions in the field of vector biology and uncover genes that play a role during pre-adult or early adult developmental stages. The main advantage of this technique is that it provides researchers with an opportunity to perform rapid functional genomic studies beginning gene knockdown during the developmental interval, the pupal stage, when RNA interference injection protocols have not been implemented in the past.
Visual demonstration of this method is critical because the injection steps can be difficult to master due to the precision required and the delicate nature of pupae during this developmental stage.
After selecting the gene of interest, identify a 200 to 800 base pair region that is predicted to have no off-target effects, and perform standard PCR amplification to obtain double-stranded RNA synthesis template flanked by a T7 promoter sequence. Then, synthesize the double-stranded RNA and clean up the products using a commercial in vitro kit. Adjust the final RNA concentration to 3 micrograms per microliter. Next, use a standard 2% agarose gel to compare 1/2 microliter of the double-stranded RNA with template DNA and standard markers. Once run out, check the double-stranded RNA band quality and length. There should be no visible non-specific products. The double-stranded RNA will migrate more slowly than template DNA. For storage, prepare double-stranded RNA